Okay, so now we will see some rules provided by other scientists based on the pairing of nitrogenous bases in DNA. Okay, so rules. We're going to see some rules. The first rule is known as base pair rule. So this is related with the double helical structure of DNA. But when question asks, draw the double helical structure of DNA, just draw that structure and explain along with it. No need to show this one, okay? These are the rules that you need to know, and it is provided by other scientists based on that double helical structure of DNA. So according to base pair rule, it says that adenine, I am going to show it with the formula only, okay? If you want to write the sentence, you can write by one. According to base pair rule, it says that the adenine always pairs with thymine by making double bond. Okay? This means double bond. And thymine always pairs with adenine by making double bond. Okay? हम लोग जो ये structure draw किया था ना, this structure, the nitrogenous bases. So actually the adenine, it pairs with thymine this way by making double bond. If adenine pairs with thymine, thymine will pairs with adenine, vice versa, okay, by making double bond. ऐसे actually ऐसे pairing होके रहते हैं. But for our concern, we have just simply drawn a straight line to understand it in easiest way, okay. But the actual structure, it is like that. So next is, and the cytosine always pairs with guanine by making triple bond. Okay, triple bond. देखो ये सब ना entrance में बहुत ही important है. Entrance में आते हैं. Adenine और thymine के साथ हमेशा double bond बन के रहते हैं, and cytosine and guanine हमेशा triple bond form होके रहते हैं एक दूसरे के साथ. Suppose this is cytosine, there is guanine, so they will form triple bond. ठीक है, triple bond form करके रहते हैं एक दूसरे के साथ. So remember, okay, this is important, you cannot forget. And if cytosine guanine, guanine will cytosine. So simple है, simple है. सिर्फ bond को याद रखना, कौन किसके साथ कितना bond form करके रहते हैं. So guanine pair with cytosine by making triple bond. So this is base pair rule. ठीक है, remember कितना bond form करते हैं. So the bond is hydrogen bond. ठीक है, जो bond बोलते हैं हम लोग bond, ये दोनों nitrogenous bases के साथ क्या बॉन्ड फॉर्म करते हैं, हाइड्रोजन बॉन्ड फॉर्म करते हैं. ठीक है, ये जो बॉन्ड है डबल बॉन्ड, ट्रिपल बॉन्ड, दिस इज एक्चुअली हाइड्रोजन बॉन्ड. वीक बॉन्ड होते हैं हाइड्रोजन बॉन्ड, कैन बी ब्रेकेबल इजीली. सो हाइड्रोजन बॉन्ड होते हैं एक दूसरे के साथ जो बॉन्ड फॉर्म करते हैं. सो कौन किसके साथ कितना बॉन्ड फॉर्म करते हैं, दैट्स यू रिमेंबर. और कौन किसके साथ बाइंड करते हैं और या फॉर्म करते हैं, you need to remember. In double helical structure of Watson and Crick, we have said that purines always pairs with pyrimidine. तो ये तो simple word में बताया गया, लेकिन actual में purine, that is adenine, it will always pairs with thymine, and the thymine it will always pairs with adenine. So purine pairs with pyrimidine. Okay, purines pair with pyrimidine. So and cytosine always pairs with guanine. So cytosine, pyrimidine, guanine क्या है, purine. So same ही है. Guanine pair with cytosine by making a triple bond. That is the only differences. कौन किसके साथ bind करते हैं और कितना bond form करते हैं, you need to remember. And the name of the bond is hydrogen bond. ठीक है. So this is about base pair rule, and there is one more rule known as Chargaff's rule or Chargaff's law. Chargaff, Chargaff's law. Chargaff's rule और Chargaff's law भी बोल सकते हैं, LAW. So according to Chargaff law, what it says? According to Chargaff law, it says that adenine is always equal to thymine and vice versa. Okay? यहाँ पे मैं equal sign दे रहा हूँ, ठीक है? This is not a double bond, this is equal sign. So according to Chargaff's law, adenine is always pairs with, not pairs, adenine is always equal to thymine, thymine is always equal to adenine. That means adenine का जितना नंबर होंगे उतना ही thymine का नंबर होंगे. Okay, suppose यहाँ से लेके यहाँ तक adenine 10 adenine we got, Okay, in one complete turn, suppose there is 1089. Okay, so try to imagine. Suppose 1089 है तो थायमिन का भी उतना ही नंबर होंगे, 89 का जितना नंबर होंगे. If there is 1089, there will be 10 thymine. Okay, if there is 10 thymine, there will be 1089. Okay, so it says the adenine is always equal to thymine, thymine is always equal to adenine. Okay, इसको slightly different manner में हम लोग ऐसा भी लिख सकते हैं, adenine divided by thymine. Okay, thymine divided by adenine, ऐसा भी लिख सकते हैं. Okay, जो जितना that means जितना adenine का नंबर होंगे उतना ही thymine का भी उतना ही नंबर होंगे. ठीक है, suppose adenine का यहाँ में 2 मिला, thymine का नंबर भी 2 मिला, so ultimately it is going to be 1:1. Okay, so the number will be equal. Understood? Okay, this is not bond. Okay, here in Chargaff law it means it is equal. देखो differences समझने की try करो, थोड़ा सा समझने की कोशिश करो. Cytosine is always pairs with guanine. Again guanine always pairs with cytosine. यहाँ पे bonding नहीं होता है. Okay, equal होता है, equal. Cytosine always equal to guanine, guanine is always equal to cytosine. देखो इतना simple है. इसको हम लोग slightly different manner में ऐसा भी लिख सकते हैं. तो जितना cytosine का नंबर होंगे guanine का भी उतना ही नंबर मिले होंगे. So this is Chargaff law. Okay, so there are two laws provided, okay, based on the double helical structure of DNA. One is base pair rule. Base pair rule says that adenine always pairs with thymine by making double bond, and thymine always pairs with adenine by making again double bond. But the cytosine pairs with guanine by making triple bond. Guanine pairs with cytosine by making triple bond. According to Chargaff's law or Chargaff's rule, adenine is always equal to thymine, thymine is always equal to adenine, guanine always. Okay, so understood? So यहाँ पे हम लोग एक problem मतलब problem question कर सकते हैं. Okay, sometimes कभी मन गया तो पूछ सकते हैं ये question. Okay, I'll give you one question. In a amount of DNA, suppose ये क्वेश्चन देखो कभी पूछ सकते हैं एग्जाम में, कभी आते हैं. So it's better we complete all. In a DNA molecule, if the amount okay adenine is given as 30%. ठीक है, अगर एक DNA molecule में adenine का amount कितना है 30% है, then what will be the amount of cytosine? ऐसा क्वेश्चन एंट्रेंस में तो आता ही है, और बोर्ड में कभी मन गया तो एग्जामिनर सेट कर सकते हैं ऐसा क्वेश्चन. सो नाउ ट्राई टू सॉल्व दिस. अभी हम लोग मैथ्स में आ गए. So in a DNA molecule, if the amount of adenine is 30%, what will the amount of cytosine? Adenine का कितना दिया हुआ है? 30% दिया हुआ है, right? 30% मतलब out of 100, तो cytosine का कितना होंगे? So according to Chargaff's law, what we know, adenine is always equal to thymine, right? Adenine is always equal to thymine. मतलब अगर adenine का 30% दिया हुआ है, तो thymine का भी 30% होंगे, क्योंकि adenine and thymine equal है, right? So अगर 30% adenine हो गया, thymine का 30% हो गया, तो cytosine का कितना होंगे? Cytosine का कितना होंगे? And we know cytosine is always equal to guanine, right? Cytosine is always equal to guanine, right? तो 30 ले लिया adenine, 30 ले लिया thymine, बचा कितना? Out of 100, 30 60 हो गया, तो 40% बचा है. So out of 40%, cytosine will take 20% and guanine will take 20%, because we know cytosine is equal to guanine. तो cytosine का वैल्यू कितना होंगे? cytosine का वैल्यू कितना होंगे? 20%. Understood? Very easy, right? Very interesting and very easy. Even the primary school student will solve this kind of problem, right? So I will give you one homework here. if in a DNA, if the amount of, इसको चेंज कर देता हूँ, समझ में आया है? एडिनिन इज ऑलवेज इक्वल टू थाइमिन, एडिनिन का जितना होंगे थाइमिन का भी उतना ही वैल्यू होंगे. तो साइटोसिन का कितना होंगे पूछा गया है, तो साइटोसिन इज ऑलवेज इक्वल टू ग्वानाइन, साइटोसिन का जितना वैल्यू होंगे ग्वानाइन का भी उतना ही वैल्यू होंगे. तो एडिनाइन इज इक्वल टू थाइमिन, एडिनाइन का 30% दिया है, तो 30% एडिनाइन हो गया, तो थाइमिन का भी 30%, एडिनाइन इक्वल टू थाइमिन, 30 हो गया 60, आउट ऑफ 100 कितना बचा 40%. तो साइटोसिन विल टेक 20, 40 में से 20 लेगा साइटोसिन और 20 लेगा ग्वानाइन, तो वैल्यू कितना हो गया साइटोसिन का 20%, राइट. ओके, आई विल गिव यू होमवर्क, प्लीज सॉल्व दिस एंड शो मी इन व्हाट्सएप. जो नहीं दिखाएगा उन लोगों को मैं ब्लॉक कर दूंगा वीडियो से, दे विल नॉट एबल टू सी माई वीडियो. आई वांट 1 मिलियन व्यूज, ओके, अदरवाइज आई विल स्टॉप डूइंग वीडियोस. सो इन ए डीएनए मॉलिक्यूल इफ द अमाउंट ऑफ चेंज कर देता हूँ मैं, साइटोसिन is 20%, what will be the value of थायमिन? इसको होमवर्क करके दिखाओ मुझे व्हाट्सएप में, ठीक है? नाउ वी विल सी द टाइप्स ऑफ डीएनए. We find various types of DNA. So the DNA that we discuss in our video lecture, that is the B-type DNA, and that is a human DNA, and that is a right-handed DNA. Okay, make it correct. I have said it left-handed. It is not left-handed. It is right-handed DNA. So make it correct. Okay. So these are some extra things we need to know. So there are various types of DNA. In basics, we will learn. Okay. So you should know all these things, though it is not in your syllabus, but still you should know. So there are different types of DNA. So first, here in this classification, we will classify DNA based on their number of base pair. Okay. So based on their number of base pairs, we will classify DNA into various types. So we will classify on the basis of their base pair, that is nitrogenous base pair. So first we'll take the types here, and then below we'll take number of base pair. Okay, base pair, this is short form. Okay, so we find A type DNA, B type DNA, C type DNA, D type DNA, and Z DNA. Okay, all this DNA, it is going to help you in your entrance portion. Okay, so the DNA that we basically talk about in human DNA are B-type DNA, and it is a right-handed DNA. Okay, the DNA moves in a clockwise direction, so we call it as a right-handed DNA, and human DNA are B-type DNA. Okay, so apart from B-type DNA, we have A DNA, C DNA, Z DNA, and D DNA. So we will just see the basic things here. Okay, so just the basic things. We will not go in details, but if you go to higher study level, you need to study all those DNA in details. So number of base pair, we classify on the basis of number of base pair. In case of A DNA, it is about 11 base pair. So the base pair, that means in one complete turn of DNA that we are talking about. The base pair, in a sense, it is in a one complete turn of DNA. When DNA make one complete turn, we have discussed, it is about 34 angstrom, and within one complete turn, it consists of 10 nitrogenous base pair. Right? We have seen that. And the distance between two nitrogenous base pair are 3.4 angstrom. Right? We have seen. That is in B DNA. But in A DNA, in one complete turn of DNA, that is in one complete helix, it contains 11 nitrogenous base pair. Okay, that's it. Just remember that. We don't have to go in details. Okay. The B DNA we already know. So in B DNA, it consists of 10 nitrogenous base pair, right? C DNA consists of 9.33 approximately base pairs. In ddNA, it consists of 8 nitrogenous base pair. In zDNA, it consists of 12 nitrogenous base pair. Okay, so try to remember the value. This is important from your entrance point of view. Okay. So the number of base pair in a sense in one complete turn of DNA. Right? We have discussed one complete turn. All right. So again, we will classify on the basis of their nature of the helix. Okay, on the basis of their nature of the helix, whether it is right-handed or left-handed. Right-handed means if DNA moves in right hand, it is clockwise. Whenever DNA moves in a clockwise direction, clockwise. So clockwise, that means it is right-handed. So we will classify again on the basis of their nature of helix. So on the basis of nature of helix, the A DNA are right-handed. All the DNA are right-handed except Z DNA. Right-handed DNA. When we say right-handed, that means the DNA moves in a clockwise direction. Clockwise, okay? So all right-handed are clockwise direction. This is also right-handed, this is also right-handed, and this will be your left-handed. So left-handed will be anti-clockwise. Anti-clockwise. I hope you all know clockwise and anti-clockwise direction, right? So the DNA which moves in a clockwise direction, we call it as a right-handed DNA, and the DNA which moves in an anti-clockwise direction, we call that DNA as a left-handed DNA. Okay. From entrance point of view, the question comes from this part, Z DNA, because this is the only DNA which shows different characters. Okay, so try to remember the number of base pairs, try to remember the nature of DNA. In my previous video, I have said that B DNA, left-handed DNA. That is not true, so make it correct, right-handed DNA. Okay, yeah, it happens sometimes because of this very hot humidity and temperature. So what else you need to know? So these are the types of DNA. Okay, so these are some extra thing I'm giving it to you, which is not included in your NCERT book, but still it's better to know everything. So next, you need to know one term. Palindrome. Okay, you need to know one term when we talk about DNA structure. Okay, palindrome. I think you might have heard this term, and this question came in this year exam, final exam in a second year for 2 marks. What is palindrome? So palindrome is a sequence of nitrogenous bases. Okay, palindrome is a sequence of nitrogenous bases, and when we read the nitrogenous bases in both the direction, we read the same. Okay, you will understand. Wait, so DNA is made up of double strand, right? And the 2 strands are anti-parallel. Anti-parallel means if one strand has 5' prime, the other strand is going to have 3', right? We say prime. Okay, the dash is there, in at the top of the number. We call it as a prime. So DNA is double-stranded. Is made up of double-stranded. Yes, it is double-stranded, and the 2 strands are anti-parallel, and they are in a helical structure. So far we have known, and that is the structure given by Watson and Crick, right? Now for our concern, to make it easy, let me draw a straight line. Okay, this is one strand of DNA. Normally the DNA is not straight, the strand is not straight, the strand is helical, but here for our concern, for our understanding, I am drawing it, I'll show it in a straight line. Okay, this is one strand, This is another strand. One strand has 5 prime. The end of that strand is going to have 3rd prime. Okay, complementary to that strand, if it is 5 prime, the other strand is going to have 3rd prime, and the end of that is going to have 5 prime. Okay, this is actually this straight line that is we are drawing, the strand is actually a pentose sugar. It is composed of a pentose sugar. Okay, because for our concern we are making it in a straight line, but actually it is a molecule pentose sugar, and it is not straight line. Actually DNA is not in a straight strand, it is a helical structure. The structure that we have drawn in our last video, this structure, right, this structure, actually this structure is actually a pentose sugar. Okay, to show it, to make it easy to understand it in easiest way, so we have drawn like this, but actually it is a pentose sugar. Okay, so don't get confused, and 2 strands are antiparallel. So in this strand, nitrogenous bases is attached, right, and purine always pairs with pyrimidine. According to base pair law or base pair rule, adenine always pairs with thymine by making double bond, and cytosine always pairs with guanine by making triple bond, and vice versa. We have already seen that. So this is actually a pentose sugar. Okay, so there will be nitrogenous bases. So just for our concern, okay, we are drawing like this. So just let's take example. There is a nitrogenous basis like A, D, C, G, C, D, G, C, D, A. Okay, here we have suppose nitrogenous bases. A, adenine, thymine, cytosine, guanine, cytosine, thymine, guanine, cytosine, thymine, adenine. So we have a long chain of nitrogenous bases along with the pentose sugar. So with the complementary to that, the other strand is going to have, if there is a adenine, okay, let me put some little bit space between those nitrogenous bases. Okay, wait, otherwise you will get confused. Okay, थोड़ा सा space देता हूँ मैं, A, D, C, G, D, C, A, so हो जाएगा. So we have this strand. So with complementary to that strand, the other strand is going to have adenine. उसका opposite में क्या होगा? Thymine. Because adenine always pairs with thymine by making double bond. So we are not going to show the bond here. Okay. Otherwise, a diagram will become very complex. This is for us to understand. Okay. So we won't show like that. So we'll simply put it like this. Okay. Bond दिखाने का जरूरी नहीं है. This is for only to understand. Okay. So adenine, you know. Okay. Adenine pair with thymine by making double bond. So thymine again it pairs with adenine. Cytosine pair with guanine, but there will be triple bond. The bond is a hydrogen bond. Okay. So we will simply keep it like that because otherwise the diagram will become very complex. So we'll keep it like that, and you already know. Okay, the bond between each other. Guanine pair with cytosine, again thymine will pair with adenine, cytosine pair with guanine, adenine pair with thymine. What this? Okay, this is the structure that we find. Okay, suppose example, we are taking example here. Now what happened when we read both the strand in opposite direction? Okay, in both the strand in opposite direction we read the same letter. Okay, when we read both the strand in opposite direction, we read the same letter. Okay. Now here in this strand we have A, T, C, G, T, C, A. Now we read it. Okay, starting from 5 prime in that strand we have A, T, C, G, T, C, A. Now read it from this strand from 5 prime. We're going to get the same letter. Okay, T, G, A, C, G, A, T. Okay, when we read this strand from 5 prime, we have this nitrogenous bases. Again, if you read from 5 prime, the opposite strand from 5 prime, when we read the letter, we get the same letter like this. Okay, we get the same letter like this. That is a palindrome sequence, we say. What is palindrome sequence? Palindromes is a sequence of nitrogenous bases. When we read in both the direction, we read the same. When we read in both the direction, we read the same. Okay, देखो adenine, thymine. Okay. Okay, wait. Okay, when we read in both the direction, we read the same. Okay, adenine, thymine, cytosine, guanine, thymine. Okay, here we have this strand. Now read it from the 5' prime. Adenine, thymine we have, cytosine, guanine we have. So this is the same. Okay, only difference is the thymine it replaced with adenine here. Okay, are you getting or not? To make it easy, let's take example like in others example. Apart from DNA strand, we can take the example like it is there in your NCERT book, Malayalam, just like that. Malayalam. So when we read Malayalam from this direction, we read Malayalam. Okay. Now if we again read from that direction, we also read the same words, Malayalam. So just like that, this is palindrome sequence, just like that only. Okay. When we read both the strand in opposite direction, we read the same. Okay. The only difference is the A got replaced with thymine. Okay. Thymine and adenine, they are equal only. Cytosine and guanine, they are equal only. Okay, because the other strand is going to have the complementary to that strand. Okay, so to make again more easy, so we take other example like Malayalam. When we read both the letters, okay, that means if we read from that side also, if we read from that side also, we read the same words, we read the same letters, just like that. Or again, if you take the example of I, so same. E-Y-E-I, if you read from that direction, I, if you read from that direction also, it becomes I. So there is no any changes. Okay, so this is palindrome sequence. I hope you understood. So in exam, you try to show this example, and this example also you can show, Malayalam also you can show. But you need to write the sentence, okay? What is palindrome sequence? Palindrome is a sequence of nitrogenous bases. When we read both the strand in opposite direction, we read the same. That's it. Okay. So this is about palindrome. Here, though the letter do not look exactly similar to that strand in here, but it is same only because G is equal to cytosine and thymine is always equal to adenine and vice versa. It is the same only. Okay. All right.